I know I've not taught you, but I just want to know what you are. So what do you know as recombinant DNA technology? I'm not expecting the perfect answer because this is what I'll be teaching you today. So and you don't need to cheat. So what is recombinant DNA technology? What can you say?
Mm.
All right, Amal, I'm waiting for you.
Um,
Do you have an idea? So we don't waste time. Do you have an idea?
mm. I'm not sure.
So Daniel,
Yes, sir.
Daniel, do you know what recombinant DNA technology is? Daniel, see guys, if I call you and you don't answer me on time, it simply means you are not with your device. You are just logging in for the class. And if you do that, I'm going to kick you out. So everybody knows how long I've been calling Daniel Hiram and he's not answering me. It simply means he's not with his device. So I have to kick him out. So Ogbonna Ikele, so do you have an idea of what have you heard the term recombinant DNA technology? Have you heard that term before?
Good morning, sir.
Good morning.
I feel it's like bringing DNA molecules like together to produce like from different species, like the joining of two different species of DNA.
The Nathaniel Fred, what have you heard the term gene cloning before?
So I can hear you.
Have you heard the term gene cloning? Have you heard it before?
Gene cloning, yes, sir.
So in what context did you hear it? And what do you think it
It's kind of a process in which a gene is located, something. I can't really, so I'm not sure. Hello, sir.
Okay, I heard you. Okay, at least. What about you, Fatbola? Have you heard the term gene cloning before? So Daniel Hiram, I called you. We waited for long. You did not answer me. So if I call you next time and you don't answer, I will not allow you entry again. So please and please, if you are for this class, this way you are and be serious. You cannot log in.
Okay,
I will not know that you are not there. So Daniel Hiram, we
okay, sir.
are, have you heard the
Huh?
term recombinant DNA technology before? Daniel Hiram,
Sir?
recombinant DNA technology. Have you heard it before?
No, sir.
Okay, Fatbola.
Sir? Sir?
So can you answer your question?
Gene cloning is like copying another gene, like copying like another.
Okay, at least. So Precious Eziri,
Sir?
have you watched? No, no. I'm calling another person. So Precious Eziri, what do you know about gene
Gene cloning, I feel, is the replication of genes to, like, maybe you want it in a massive, maybe like for a virus, I don't know, maybe like a virus, you want it, that's the way I heard it. Like a virus now, you want it to like be many or much, even know the genes. So I think they clone it. Sir, can you hear me?
Yes, I can hear you.
So I feel is when they want to replicate a gene, like want to replicate that they do the cloning, or or maybe something, I don't know. Or replicate a human being, I don't know why they clone a human
Okay, thank you very much. So, guys, I would really advise you guys to please and please um, try and simulate as if you are in class. Be attentive. Um, be open-minded. Know that if you want to learn this thing, you can learn it. You can understand it, and irrespective of where you are, uh, just ensure that where you are is silent, and you are also open-minded. And so I was trying to understand what you guys know so that I will be able to know the languages that I was speaking. And I'm not looking at the situation where you will not be able to understand what I'm saying. So that was why we had a pre-class assessment. So before I start going into everything that we're expected to talk about today, I want to provide some background information. So when we talk about in the context of when some people hear the word DNA, genetics, biotechnology, one of the controversial aspects is that they believe that we are trying to produce another human being. And if you've been watching all these science movies, you will see when they will say that, oh, they went to the lab, they produced an entirely different human being. And so the person now became abnormal. The person was not the new organism that was created, became, uh, for instance, things like Godzilla, things like the Jurassic Park, all those kind of things that they usually attribute the origin uh, of the organism or of the gigantic animal that is terrorizing people to a particular laboratory. And in more recent times, especially what we are dealing with in the present time, I'm talking about the pandemic. Uh, there are some schools of thought that is even some politicians are actively driving uh, the notion that the COVID-19, the causative organism for COVID-19, which is the SARS-CoV-2, was genetically produced in a laboratory in Wuhan in China and was now released into the general population. And giving that notion that human beings were genetically modified, or that the virus too was created in a lab and was released into the environment. So the my goal today is to let you know the facts uh, and the science of how stuff like this can potentially be done in the laboratory. How um, we are going to look at the pros and cons, the advantages, and why this is important. When you look at recombinant DNA technology, we are looking at a means through which one of the easiest examples that I love mentioning is the example of the rice that we consume every day. In the past, rice was just blank rice. It was rice and nothing more. But these days, we see rice that are fortified with vitamins, rice that contain vitamin E, vitamin D, and some other essential materials. And so, what is the advantage of this? The goal is that even if you travel abroad, if you've been outside the country, you know that they eat a lot of cereals. Cereal foods are one of the most popular foods that people consume outside this country. And you just see that they are going to pour it into a bowl, and they will add milk, and that is enough for a breakfast. And when you look at the children are always good looking. They are looking more healthy than we that we are going to eat a mountain top of rice. You put stew somewhere. You have to add beans. You still say you want to add for you to take a balanced diet. You want to take you want to add other essential things. So by the time you are eating, your stomach becomes very, very full. You cannot quickly, you cannot, in an attempt to take balanced diet, you take a lot of food because these essential nutrients that ought to be present in the balanced diet, they are present, we are taking them from different sources. But with biotechnology, food alone, a single bowl of cereal is made to contain everything that a baby needs, that a person needs for energy, for growth, for the immune system in that single bowl. And that is one of the major applications of biotechnology. In Nigeria too, I'm in Ibadan, and there is this popular research institute that is called the IITA. So Nathaniel, have you heard of IITA before?
No, sir.
Fatbola, have you heard of IITA before?
No, sir. No, sir.
You've not heard of IITA. Who has heard of IITA before? Allow Damola, do you know what IITA is?
Yes, sir.
So what do you know about them?
I know that they genetically modify fruits also. Their fruits are different.
Let me be sure that you know them. Give me an example of a food that they genetically modified so that I'm really sure you know them.
Like, I think orange, yes.
Okay, at least you know it. Precious Eziri. Are you there?
Yes, sir. Yes, sir.
Do you know IITA?
No, sir.
Okay. Now, IITA simply stands for the International Institute for Tropical Agriculture. So they carry out agricultural researches. And one of our staple foods in Nigeria is cassava. When you want to make eba, you want to make amala, when most of these are from cassava. So Nigeria and several other African countries, we consume a lot of these of cassava almost on a daily basis. There are some people that even eat cassava twice in a day. Now imagine if the cassava is made to contain all the essential nutrients that somebody needs. They are able to squeeze in some vitamins. Don't forget the vitamin E, the vitamin D. And as we are consuming our normal cassava on a daily basis, we are also getting these nutrients from that cassava, which means instead of having amala, then you have bakery, you have a bean somewhere for you to be able to, you also have to throw in some vegetables from that cassava alone. The cassava contains everything, every nutrient that you need for good functioning. So that is what they do here. They fortify cassava, they fortify other agricultural produces so that when we are consuming a bowl of food, we are actually getting all our essential nutrients in one meal. And it's something that is happening right here in Ibadan. It's not something that is foreign, something that we don't know about, something that people that is still like all those movies. It's already happening on a daily basis. Another example that I want to give is the oil, the granite oil. If you look at the granite oil, it contains before. If you know how they used to make granite oil, it wasn't packaged in the way that it is being packaged these days. And one of the in the race to capture a significant market share, what the granite manufacturing companies are doing is they are adding a lot of essential nutrients, a lot of important stuff into that granite oil, so that when you pour the granite oil, you are getting your vitamin D, you are getting the vitamin C. Look at the salt too. All this Dangote salt. It's no longer just salt. It comes with iodine. Iodine is loaded with that salt, and there is also addition of extra stuff in it. Then probably the last example that I want to give has to do with farm produce. Sorry, this animal produce. Um, there is a good example I will give to you is that we have different types of chickens in the market. There is the conventional and popular local chicken and the broilers that that they have everywhere. So I want to ask Daniel Hirim, what's the difference between, what do you see as a difference between the local chicken and the other types of chicken, the broilers? So what, personally, what do you see in that is different? Daniel,
Sir, come again.
what did you say?
Please come
What do
again.
you see as the difference between the local chicken and other types of chicken?
Vocal chipping and other
The
types of
local
chipping.
chicken, uh, do you know the difference? Can you see any difference?
No, sir.
Ekele Ogbonna,
Yes, sir.
what's the difference? Local chicken and other chicken.
Sir, I feel personally maybe you can easily alter the chicken that is not the local chicken. It's like it's hard for you to alter the local chicken, like the ingredients of local. Personally, I think that's what I feel.
I'm not talking about genes. I'm not talking about, forget genetics, forget this class. Local chicken, if you see a chicken, the typical local chicken roaming around, and the other types of chickens that are big. So what do you see? What is different in them?
To me, I feel the local chickens are not as healthy as the other chickens.
How do you know they are not as healthy? What do you see?
Like I feel the other chickens actually cared for more than the local chickens. Like they tend to give them food that are trendy and like tend to treat them better.
So Fatbola, you should know this very well. So local chicken. Have you seen a local chicken before? Madam Olakabola.
Yes, sir.
So you should. What's the difference?
The local chicken, they don't have, they're always looking skinny. Like they don't have, they're always big and mm, as
Yeah, you
compared
know,
to the
okay,
bread.
Sir, can I say something?
please, yes, please.
Yeah, concerning the local chicken, for me, what I feel is that the local chicken and the English chicken, the local chicken, for me, they're actually more healthier than the English one.
How? Why?
The local chicken is very rare for them for you to see that the local chicken falls sick.
Okay. Now Let's talk about marketing and product packaging. Which one looks better, Daniel? Which one do you think is looking more beautiful? The local chicken or the broilers?
Mm, for me, it's if you go to that aspect, you look at the foreign chicken because it has the beauty. But
Which one is
when looking at
which
the
one is
local chicken, it has the I would say it's more nutritious more than the foreign because the foreign they use chemicals sometimes to feed them.
okay? So what about size? Which one do you think is bigger, local chicken on the or the broilers?
The foreign chicken is bigger than the local chicken.
Okay, great. Now, if you are a business person, you want to sell chicken, you want to sell at a very high price, you want to make profit. Um, Nathaniel, which one do you think a smart business person would be presenting or would be selling? The local chickens or the ones that are bigger and are looking more healthier? So, what do you think?
I feel if um a businessman wants to get something like that, they will go for the bigger one because the size, most customers they look at the size for getting something and how heavy it is.
So yeah, that is very correct. It is something that is normal in every agriculture. There is also, I can go on and on. We have tomatoes. We have some tomatoes that look at how we produce tomatoes in Nigeria. Most of the tomatoes are from the north, but they are selling tomatoes in the southwest and the southeast. So let's say they are transporting tomatoes from Kano and they want to bring it down here to Ibadan. And so in the process of it, I was reading something that said like 50% of farm produce, of tomato produce, perishes in transit. Because by the time they are packing them up in the big lorries, they will be collapsing and they will be melting away and destroying. They will be perishing. But we have some brands of tomato that when they, and the reason why this is happening, why they are perishing, is because the outermost skin of the tomato is very weak, so it cannot withstand the pressure. So what they did in biotechnology is to genetically modify the outermost skin of the tomato to make it thicker so that it can withstand more pressure. And that reduces strategically and significantly reduce the loss due to in transit. And maybe the last example I will use, because we have lots of them around us, is the onion. There are some onions that when you slice them, tears will be coming out of your eye. The whole types of onion. I think some of them still exist today. But we now have some onions that even when you cut them, even if you squeeze the content of the onion in your eyes, you won't feel anything. What they did was that there is a gene that is responsible for that teary eye, for that sensation. So when in the process of genetic modification, they were able to silence or modify that particular gene. So those are there are lots and lots of examples that we can use for you guys to understand it. But I just want you to know that it's not something that is not heard of. It's something that we are already dealing with on a particular daily basis. So today we'll be looking at three different issues. We'll be looking at, and I will try as much as possible to go as far as I can, but I also attempt not to overstress you guys so that you're able to understand to a large extent what I'm talking about. So the three issues we'll be looking at include our recombinant DNA technology, we'll be looking at cloning, and we'll be looking at genetic biomarkers. So so I want to go from the angle of recombinant DNA technology. So when we talk about recombinant DNA technology, it's from the term I always advise students to understand the building elements, the words that are making up the terminology that you are trying to define. So when we talk about something is recombinant, it's from the word recombination. It means there is an original one, but we've recombined it. So it simply means that if there is a feature that we don't want, it simply means that we've removed that feature. And if there is a feature that we desire, what we are talking about here is that we've actually we've ensured that that thing that we don't want has been removed, and the feature and or the characteristic or the character that we desire, we've introduced it. I always like using some forms of natural, although non-scientific explanation to do it. If a lady is tall, so if a lady is very short, one of the things that Nigerian women many women will talk about is that I cannot be short and I will still marry somebody that is short. So they look for a very tall person to marry, and already it simply means you're already doing some forms of recombination because we believe that the genes for the person that is tall is going to rub off on you. You will combine your own genes, and you'll be able to have tall children. So, but in the laboratory, this is also what we are trying to simulate in the laboratory. And all of these three issues that I mentioned, the cloning, the recombinant DNA technology, and the markers, they are captured under a section of science that is called biotechnology. So when you define biotechnology, biotechnology is a method through which a living organism is used to change or to incorporate a particular character into another living organism. So using the example of somebody that is tall, not wanting to have very tall children. So you are expected that okay, that person that has the desired character is going to we are going to introduce the genes from that person into the person that does not have the character, so that the desired character is acquired. So what we do, the way we carry that out is that we apply scientific principles to the processing of materials by biological agents. It's just a simulation in the laboratory of what happens naturally. You know, when you want to have children, when you want to raise kids, you go with the natural way. God is the one that does all the assortments and gives you what He desires, He proposes in mind to give unto you. When you recombine DNA technology, we are the ones that we are trying to play God there. We are trying to say, this is what I want. What I want, I put it in this organism. What I don't want, I take it out. And it's a very scientific way. So, by definition, genetic recombination, what we are doing is that we are exchanging information between two DNA segments. So, don't forget, if you've been following this class already, you should be able to know what is the difference between the DNA. You should know what a gene is, and you should know what a chromosome is. So DNA is a genetic macromolecule. It's a chemical substance because it's an acid. So the oxyribonucleic acid, it occurs as a double helix strand. And on the DNA, we have regions that code for something, which are called the genes. So what those characters, whether it's tallness, whether it's color, whether it's whatever character that we desire, there is a particular gene that codes for it. So if we want a particular organism to have a desired character, what we are going to do is that we are going to take the gene that codes for that character in an organism that has that character. We are now going to take that gene and the area where that gene is located, which is the reason why we call it the DNA segment. The area where the gene is located, we are going to remove it from the person that has, from the organism that has it, and insert it into the DNA of the organism that we want to see that character manifest. Nathaniel, does that make sense to you?
Yes, sir.
So can you recapture what I said about how we go about genetic recombination?
Okay. Well, you said whatsoever trait you want to extract from an organism, you extract it, then you replace it with the you replace it with the DNA of the other one that you are using.
How you, Damola, does that what can you help him out?
So you said you take the needed gene from the organism and um, place it into the desired organism that you want to like give that gene to.
What about you, Precious and Zuri? Let me know what you know. Precious, are you with us?
Sir, yes, I'm with you, sir. Don't you? No, no. Mm-mm.
I'm listening to you.
So it's only the sir.
How does genetic recombination
Sir, I
work?
can't hear you.
How does genetic recombination work?
As she said, oh, okay. I said, as she said, I feel that it's still the same thing. Maybe you replace one with another one, or you have one to another one. I don't know. I'm not sure.
Okay, please mute your mic and at least you guys have some idea of what I'm talking about here. So we are exchanging information between two DNA segments. There is one of the segments is from the organism that contains the desired character, desired trait. The other segment is for the organism that does not have that character, but that we want it to have that character. So those are the two DNA segments. For instance, now I'm not sure if Nathaniel is a very tall guy, and we want the but what is her name? But Precious is not too tall. So and Precious desires to have babies that are tall. If we want to do DNA recombination, what we will do is that we are going to take a DNA of Nathaniel that is very, very tall and insert it into the DNA segment of Precious that is not tall. So that region that contains the DNA that for the height determining region in Precious's DNA will be removed, and we will now replace it with the height with the height determining region in Nathaniel's DNA, so that when everything comes together, Precious now has the gene for height and everything is okay. So in a nutshell, that is what we are essentially doing in genetic recombination. So even though this happens in, like we said, naturally, sometimes it can happen naturally, but we can artificially do it. The reason why it's called recombinant DNA technology is because it's an artificial means through which a gene of one species is transferred to another living organism. So you can still, if you want to talk about it again, you can talk about it in the context of genetic engineering. So by the time we are done, by the time we have completed the process of recombinant DNA technology, the product will now be a unique DNA molecule that is different, that is no longer the original source. Because if you understand how patterns of inheritance work, normally for you to have a particular trait, what you need to do is that you get one from you have a total. Every one of you has a total of 46 chromosomes. 22 of which are autosomes, which are chromosomes 1 to 22. And so 22 times 2, that makes it 44. So the remaining two are for your sex chromosomes. If you are a genetic male, you are going to have one X chromosome and one Y chromosome. If you are a genetic female, you are going to have two X chromosomes. Now, when this happens, that is the normal way. But the reason why you are having two copies is that you are getting, let's look at an example of chromosome 1. So you are having two copies of chromosome 1 because you got one chromosome 1 from your dad and one chromosome 2 and one chromosome 1 from your mom. So the two comes together and they give you that trait. So because you are getting it, that can be seen as a natural way through which recombination can happen. But it has already been done for you. This was not influenced directly. But in recombinant DNA technology is an artificial means through which we achieve what God does naturally because of our desire to have a particular trait in a particular organism for whatever need that we may want it for. So, but in this case, so like I said, it's an artificial method. Recombinant DNA technology is a highly artificial method. And so what we are doing is that we are joining DNA fragments together. We are joining DNA fragments together. And these DNA fragments are usually from different biological sources, different organisms, but we want them to have the particular trait. So if you want to define it, it's a series of procedures that are used to recombine DNA segments and the reason is this, even though the DNA is a double helix, they studied the DNA very well. It has been studied extensively. And we know that under certain conditions, when we put some conditions together, a DNA molecule, a recombinant DNA molecule can be introduced into a particular cell. That thing that we've done in the petri dish, that recombination that we've done, when we've completed it, we know that the DNA in the cell, we can actually open them up and it can now accept what we've produced. And that will now, the thing that was produced in the laboratory, when it is reintroduced into the cell that we want to see those characters, those recombined DNA molecule will now undergo replication and will start producing that particular feature. And you may want to ask that why is this something that we need to, you may want this medical laboratory science, not just agriculture. You can ask yourself, why is this important? Why will we be even be thinking of applying this? So there are a lot of applications, potential applications in healthcare. So Precious Eziri, how do you think, no matter how theoretical that you know, what are the potentials of this uh, innovation, of this technology in science and in healthcare? How do you think we can apply it in healthcare? Precious. Precious, are you there?
Sir.
Answer my question.
Yes, yeah. I can't.
Answer my, you were not on your
Sir?
feet because you did not get the question, Abby.
Sir? Sir?
Who were you? Did you get my question?
Yes, I was. Yeah, I was here.
To answer my question now.
No, sir. Sir, I didn't get your question, sir.
Why? So you are not even
That was
concentrating
yeah.
with what I'm. Araoye Damola, did you get my question? How can we apply this in healthcare?
Um, I'm not sure.
I
I think
understand. I just want
maybe in crime scenes or so. I'm not sure.
Nathaniel.
Hello, sir.
So how do you think we can apply it? So if any one of you is busy with something else, just leave my class, please. Precious is really that I will ask you a question and you not answer it. I cannot tolerate that. I intentionally sent you out. So if you are here, make sure you are fully here. If you are not fully here, you don't even bother being with me. So Nathaniel, how do you think we can apply it?
So I think we can. Uh, so I really don't have idea about it. Well, I was thinking crime scene also.
No. Okay. One of the possible ways is that look at a particular disease that is called sickle cell disease. So Oyinda Mola, what happens in sickle cell disease?
Sir,
Answer my question.
the like breakdown of red blood cell. So I'm not sure, but I think it's the breakdown of red blood cell.
What do you think causes sickle cell disease?
Sir, can I answer?
Oyinda Mola, what causes sickle cell disease?
So I don't know.
Precious, what causes sickle cell disease?
Sir, the shape in the I feel is from what I saw, the shape in the blood is different from the shape of SS patients is of their red cells are different from the shape of normal AAS people. even AS have the same, AS have a little of that same shape, but in SS it's more abundant. So whenever I feel whenever the blood wants to move, the shape is like is always squeezed. I don't know how to say it very well, sir, but that is all I feel is just like one of the things, just like the shape in their red cells.
What is responsible for the abnormality that you just described? What is causing it?
So I think
I feel it's
Nathaniel, Nathaniel, we are waiting for you.
oh okay, so I think it's a mutation in the gene mutation.
So there is a mutation that is responsible for a change in amino acid, and that amino acid is responsible for the features that Precious described. So when we know, so a theoretical way of solving sickle cell disease problem would be for us to just go in into the DNA of the patient and correct that mutation and replace it with the normal uh, nucleotides that it ought to be. But so that would be if we are able to achieve that, we are going to improve the lives of, we are going to technically and summarily cure sickle cell disease. And there are several other diseases like that too. For instance, in most of the cancers, in many cancers, as a result of the mutation in the genes that regulate cell replication and and the gene that stops cell multiplication. So, because there is a mutation in these genes, cell replication will be going on at an abnormal rate, nothing to stop it. And the mechanism to correct the errors, they are already error prone themselves, so they are already not functioning. So what the way we can cure cancer would also be that we are going to identify the mutation theoretically. What ought to be there, we are going to insert it. What ought not to be there, we are going to insert it. But that is how we can do it theoretically. So those are the potential applications of this particular disease. And we also have conditions like in insulin, sorry, in diabetes, when the cells become insensitive to insulin, they are not recognizing insulin. And don't forget, insulin is a protein, so which means there are genes that code for insulin. So which means that we can actually work on that if the production, if the cells that contain that gene are the ones that are abnormal, that have undergone mutation. We can also go into those particular cells if we can do it and remove those particular errors and replace them with what was missing. So those are the potential applications of recombinant DNA technology in healthcare. But the question you can ask is that why have we not done that? The reason why we've not done that is because of one of the reasons is because of the complex nature of the human genome. Don't forget, because I won't assume at this stage that you know, every nucleated cell in the human body contains the genetic information for that person. And the human body contains billions of cells, if not even trillions. We have at least billions of cells in us. So we have to now start checking individual cells and correcting the errors in them. So before we can convincingly say that we've solved that problem. So because if you can only do for, and of course, there are many checks and balances, there are many obstacles that you have to overcome before you can start dealing with the human specific DNA itself. And who is going to, there are also been a lot of regulations and legislations that still bans the application of recombinant DNA technology in human beings. Nathaniel, do you have a note there? I hope you guys are writing so that you will not say that I did not mention it. So please and please be taking notes as I'm giving you these examples. So lots of things from the science of human genome recombination is very, very complex. And on the other side of the spectrum too, the society is still not ready for it. And most of the countries of the world have agreed that recombinant genetic technology should be banned in human beings because while you are trying to attempt to control a particular error, those processes may be introducing a new set of errors. Although we know that the benefits are important, for instance, Nigeria and several other tropical African countries, we have a malaria crisis. Malaria is everywhere. One of the ways that they are trying to deal with malaria is, the mosquitoes that are the vectors of malaria. There are two approaches that they are using. They are genetically modifying the receptors that the malaria parasite will bind to, so that it will be unable to actually transport malaria. So they are going to mutate. Some laboratories are inducing mutation in the genes that code for the receptor that binds the malaria parasite, such that the mosquito that is biting us will be incapable of transmitting malaria. So that option is there. And even though a lot of studies, even though they've been able to produce those mosquitoes, uh, a, an ideal way to deploy that condition is to first eradicate the mosquitoes that we currently have, and now introduce the ones that would not be able to transmit malaria. So now, Nathaniel, what do you think potential problems would be if that was done? Theoretically, I'm not talking about practical, whether you know the specifics. How do you think that would be a major problem? What problem do you think we can see?
Mm, um, so I don't have any idea.
Orinda Mola,
Sir, please, can you come again?
why now you guys are not concentrating? You are not fully concentrating.
No, sir,
Precious,
I didn't hear you
precious.
well.
Sir,
Another question.
sir, I feel that theoretically the problem and challenges that we may face if we want to try that kind of thing is maybe due to the fact that it's not, I don't think it's possible actually to actually eradicate all the old mosquito that we have now, because some of them are giving birth day by day and everything. So I don't think it's really possible. I don't know how they want to go about it. And the fact that this country is not even going to help matters. So our country is not that neat to eradicate such.
Okay, that is really really. There are a lot of things. For instance, now if you are introducing these new mosquitoes, what problems do they have? The receptors that you modified, even though it will not be able to carry malaria parasite again, what if it will not be able, the mutation will make it capable of transporting other diseases, let's say HIV. If it's not able to transmit or transport malaria, what if it's now able, the mutation in that receptor makes it possible for it to transmit HIV? So there is a million and one, there are a million and one diseases that you attempt to stop one with a particular solution, biotechnology solution, we may be introducing an entirely different set of problems already. What of the interaction between the normal mosquitoes and the new ones? If they mate, uh, how is it going to be? Are they going to actually die off? So those are the questions that we may not be able to answer in our laboratory setting, in the things that we do in the laboratory. But like we said, we've seen advanced application of this particular innovation in present day practice, especially agriculture. Which leads me to, so that when we are doing recombination DNA technology, what we are trying to do is that we are introducing, like I said, uh, DNA segments from a particular, there is a particular character or there is a particular trait that we desire in an organism, that for an organism to have. So what we now do is that the gene, we are going to take the DNA segment from an organism that has that trait. The gene that codes for that trait, the segment where the gene is located, we are going to remove it. And we are now going to introduce it into the organism that we want to have that. And so what we do when we are doing that is we play around with some basic understanding of the technology that we have been presented with. Knowing understanding you guys' limitations and consciously attempting to ensure that I don't overburden you with or give you with too much load of information. But you guys really need to be so that we can bring you at par with current trends. So the first steps of the combination DNA technology were carried out on prokaryotes. So Nathaniel, what are prokaryotes?
Hello, sir.
I'm listening. Answer the question.
There are cellular organisms that lack membrane-bound nucleus.
Mola, what are prokaryotes?
They are single cell organisms that lack membrane.
So Precious, what is the difference between prokaryotes and eukaryotes?
Prokaryotes, eukaryotes are membrane-bound organelles, while prokaryotes are non-membrane-bound organelles.
What other difference can anybody or anyone of you mention? Any other difference apart from the membrane? Because membrane is not the only difference between prokaryotes and eukaryotes. Precious, what other difference can you mention? Nathaniel. Okay, speak, Precious. Precious speaks, please. Then Nathaniel takes Gloria from us. Precious, I'm listening to you.
I was talking.
Mola.
Sir,
Yeah.
you
What do you want to say?
eukaryotes, they have nucleus. Why prokaryotes, they don't have nucleus.
What other difference can they mention? What other difference can you guys talk about? Nathaniel and Damola, you guys have been quiet.
I'm here.
I think
Nathaniel,
prokaryotes. Oh, okay.
Okay, no, Damola, continue, please. Damola, continue, please.
Prokaryotes have complex cell wall, while eukaryotes are just like simple cell walls.
So which one do you think is more advanced, eukaryotic cells or prokaryotic cells? Nathaniel. Nathaniel,
Yes, sir. I can hear you, sir.
answer the question.
I feel eukaryotes is the answer.
What is the answer? What is the question?
You said which of them is which of them is more preferable?
That was not my question. Damola, so you guys are not concentrating. That's why I teach. I call you, so if you are dancing up and down, I want to be the question.
Sir?
What is the answer?
Eukaryotes.
Why eukaryotes?
Because of, so I'm not sure, but the difference is they have their membrane and they have nucleus. They have complex membrane and nucleus. So I'm not sure.
So which one do you think is easier for us to handle in recombination DNA technology? What are prokaryotes easier to work with or eukaryotes?
Prokaryotes.
Why why do you think prokaryotes are easier for us to work with in the recombination DNA technology and not eukaryotes? Ekele Bona, thanks for joining us.
No, sir, my network was having issues.
So, what is the answer to the question I just asked?
So I don't really get your question.
Why do you think prokaryotes are easier for us to handle in recombinant DNA technology and not eukaryotes? Do you know what prokaryotes and eukaryotes are?
Yes, sir. I think prokaryotes they have a single cell.
Say, answer a question with more enthusiasm. You guys are not reading, man. You are not reading. You are not even only if you cannot convincingly answer these simple questions, there is problem. You guys are not reading. You need to read well, please and please. So prokaryotes, they are very very
Prokaryotes,
simple. They are very very basic. They don't have all the mechanisms, all the barriers that many, if you want to work with eukaryotic cells or eukaryotic genomes, there are many barriers you have to overcome. There are many check, check and balances that are in place to make it almost impossible for you to work uh, with the genomics. So what we do is this, but for prokaryotes, they are simpler. Even there is no nucleus, the nucleic material is present in the cytoplasm. It's just there, so you can easily access it. Unlike when you want to do something with a eukaryotic cell, that you first have to deal with the cell membrane, get into the cytosol, go into the nucleus. If there is a nucleolus, if the chromosome is tightly packed, all those barriers that you have to overcome before you get close proximity with the nucleic material. Nathaniel, does that my explanation make sense to you? Do you understand my explanation?
Yes, I was not too clear. I didn't really.
So which part did you get so that I can know where to explain? Shed more light on.
The part where you said, where I understood it to, it's the part you said, because of the absence of nucleus in the prokaryotes, that's what makes the prokaryote more better for the recombination.
Okay, so that's one of the things. There are many things that you have to overcome if you want to work with the eukaryotic cells. And if you don't overcome these barriers, numerous barriers, there are more barriers for eukaryotic cells than prokaryotic cells. That was the reason why they started with eukaryotic cells. But it doesn't mean that they cannot work with eukaryotic cells. Science has advanced, tools have been developed, new tools have been developed so that we can actually locate various regions in the and we can work with whatever we are trying to run. So there is a combination of tools that we use, and one of them is restriction enzymes. Restriction enzymes. Have you, Nathaniel? Have you been taught restriction enzymes before?
So I didn't get you.
Do you know what restriction enzymes are?
No, sir.
Okay. Remember the DNA. Who is my good friend again? Olinda Mola. Are you there? Five
Yes,
seconds
sir.
rule. Okay, so have you
Yes,
seen the structure of the DNA before? Have you seen the DNA?
yes, sir.
So when you have the DNA, there are some enzymes that can cut the DNA at specific sites, at a specific location. There are some enzymes that can cut them. So remember I said the reason why we can do recombination is that we have a way of opening up the genome. So the restriction enzyme acts as scissors So the way it works is that we use our scissors, the restriction enzyme, to cut open the DNA in especially the regions where we want to remove. For instance, now look at let's say if the DNA from the cell that has that character that we desire, we are going to cut it open, and that region we are going to take it. We are going to use the same enzyme again. The cell that we want, the DNA that we want to insert what we have, we use the DNA, the scissors in this case, which is still serving as, which is still the restriction enzyme. We cut it open again. So the one we don't have, we remove it. So that the one that we now have, that we are holding, will now go and replace the one that we've removed. So does that make sense to you?
Sir, please can you come again?
So this is what I'm trying to say. So the restriction enzymes, they work as scissors to open up, to cut specific sections of the DNA. Because don't forget, when you are looking at the DNA, you won't be able to see where is the point that you want to replace. But because of our understanding of how genes have specific locations, every gene has a specific location. So what we do is that we look for a scissors, in this case, restriction enzyme, that we can use to cut open that particular area. It's just like if a surgeon wants to conduct a particular surgery, they will use a knife. If the surgery will happen in the stomach region, they will not be cutting the neck, because what they are trying to replace is not on the neck, it's in the stomach area. So what they do is that they use their knife to cut open the stomach area where they want to work with. And they are able to do it because they have eyes. In DNA, in recombination DNA technology, our eyes that will tell us that will guide us to where we should carry out the procedure, they are the restriction enzymes. So what the restriction enzyme does is that every restriction enzyme has a specific region that it is going to cut. So the restriction enzyme for the region that we want to cut is the restriction enzyme that we are going to introduce. So what the restriction enzyme does is that if this particular region is the area where the desired character is, the restriction enzyme is going to cut that area. And when that area has been cut, that DNA fragment that we have, we are also where we want to take it to. We are taking it from somewhere. The DNA fragment that contains the genes for the desired trait. We use the restriction enzyme to cut it from the DNA of the organism that has that character. We are now going to use the restriction enzyme again to cut open the area where we are transferring it to. So that once we've removed the one that we don't want, the restriction enzyme, we remove it here. Don't forget, look at my hand. This hand contains the DNA fragment that we desire. We want it in this organism. This is one organism that doesn't have the character. This organism has the character. So what we are doing is that with the aid of the restriction enzyme, we are cutting open the DNA fragment in the area where, in the DNA from the organism that contains it. So what we use to cut it out is the restriction enzyme. So we've cut it, and this is what I'm holding in my hand. So we are now going to use a restriction enzyme to cut open that same region in the DNA of the organism that we want that character. This organism does not have that character. So we use a restriction enzyme to cut it open. We remove that DNA fragment that doesn't have the character. Now that area is empty. So the one that we've now cut open, we will now transfer it into this particular DNA that no longer has that particular DNA fragment. So which means that the DNA that contains the genes that code for the desired character has now been recombined and is now part of the genetic component, the DNA of the organism that was not having it previously. So that one that we've now made is the recombined DNA. Damola, do you understand what I just described?
So, so I'm confused.
Explain what you know so that I can clarify it.
I understand. He said that restriction enzymes used to is will be used to cut a particular region on the organism that has the DNA. Like I was mixing it up, and he said another restriction enzyme will be used to cut open the other organism without the DNA, and I don't know. I didn't get that part well when he said you carried you like DNA will be brought into the other organism without the. I do not understand it well, sir.
Hello, Damola. What did you get?
Um, so you said that the restriction enzyme will be used to cut a part in the DNA, that the gene that is needed in a part of DNA, and the restriction enzyme will also be taken to a DNA that is needed. I don't know how to explain it, sure.
Explain it to your own language. Don't worry. So that's what you write in exam. So we know what you're writing. Exactly.
And the restriction enzyme will also be taken to another DNA that needs that specific gene, and it will cut that part out also and introduce the needed gene to it.
What about in Nathaniel?
So what I got from the explanation was you said the restriction enzyme they act as scissors to cut specific section of the DNA. So the and you said it's the restriction enzyme is more or less like our eyes, unlike those doctor when they want to carry out their operation they use their eye, but like for this particular recombination that the restriction enzyme is acting as the eyes.
Continue. So what happens? We have the high. So what do you do with the high?
So it cut out the area which which want to be extracted from a particular DNA.
Continue. Yeah, writing.
Sorry, I'm confused. I didn't really understand it very well today.
Precious Eziri, where are you?
Yeah, sir.
So let's hear your own tale.
I'm
Tell
here,
us
sir.
first. So what are we doing in recombination DNA technology?
Sir, I feel
How,
concerning that restriction enzyme. Sir,
continue. I'm listening to you.
sir, sir, can you hear me? Okay, sir. I
Yes.
feel that restriction enzyme is just like as they said, it's like the scissors that cut through some of the barriers that I don't know, maybe that cut through some from my own listening, that cut through the barriers of to get to the DNA that you're after.
So after getting to the DNA we are
I
after,
think it's
what
that
do you do?
restriction enzymes that will allow you to see through some of those barriers within there.
Okay.
Like that place I
So
don't understand, sir.
in a nutshell, what we are talking about is this. Um, I'm not sure I have the right prop to use for you guys. So please and please calm down. Stop any distraction that you are dealing with there and listening. And listen, let me summarize it. So we have this my hand is the perfect organism that has the character that we desire. This other hand is the organism that does not have the character that we desire. So what we want to do is that we want this particular organism to have the character that this one has. How do we go about it? There is a particular, there are genes that code for specific traits, and these genes are located in a particular region of the DNA. And we know we have information on the address of this particular gene that codes for what we want. So let's say this particular organism has blue eyes, but this organism does not have, it has white eyes, and we want it to have blue eyes. So what we are going to do is that we are going to take, we are going to know, first of all, we are going to know that where is the gene that codes for eye color? Where is the gene what is the location of the gene that codes for eye color that this one has? And if you enter, other you go now, okay, the genes for eye color, you are going to know where it is. So let's say the gene that codes for eye color is on chromosome 2. Is on the short arm of chromosome 2. So what we are going to do is that we are now going to look for a restriction enzyme that cuts that region on the short arm of chromosome 2 where the gene for eye color is located. So what happens is that once we know the restriction enzyme that can cut that particular region, we introduce the restriction enzyme into the DNA of that particular organism. And the restriction enzyme will now go straight to the point where we want it to cut. It's going to cut it. So which means that cut area will not be wasted, will not be destroyed. It will be present in the system, in the container that we put it. All these things are fluid, so they are apparently in fluid nature. So we've introduced it. It has cleaved it. It has cut it. So what we are now going to do is that because that region that contains the desired trait, that contains the gene for eye color, we are now going to pick it and introduce it into the same region on the genome of this particular organism that has white color but we desire blue color. But this one is intact. We also need to open it up so that we can remove the DNA fragment for eye color and replace the DNA fragment for eye color with the one that is now blue, so that this one will now have blue in color. So what we do is that we also do the same thing, introduce restriction enzymes that are going to cut that particular region that does not have the desired eye color. So we cut it, that will be discarded. It will now be replaced with the DNA fragment that contains the genes for blue color that will now come into the organism that we want it to have. Nathaniel, does that make sense to you? Do you understand it better now?
Yes, yes.
What are you still struggling with?
Hello, sir.
I'm hearing you.
I say it's getting clearer.
If I'm here and you don't get it, how do you think you are going to get it when you are on your own?
Sir, I've been following you, sir.
So do you understand what I've described?
Yes, sir, I do.
So let me show you. I think I have a demonstration, a chart that describes this. So everybody look at your screen. Daniel, what are you seeing on your screen? Precious Eziri, what are you looking at? What are you seeing on your screen?
So I'm seeing it's not really clear, but I think donor DNA, plasmid DNA, restriction, Endonuclear nucleus.
Okay, now, so this is a pictorial representation of what I've been explaining to you guys. So the donor DNA, can you see the red region is the region where the gene of interest is. So that is the part that we want. And we want it to be in this particular cell. We want that this particular cell to have that character that the region that the DNA piece that we want contains. So what they are not doing is that you can see that on its own, the donor DNA is intact, it's operating. The region that of interest is just the part of the DNA. We want to remove that particular region of interest and transport it into the cell of that particular organism that we want it to have that character. So what they are using is that they are using a restriction enzyme. The restriction enzyme they are using is called a restriction endonuclease, the name of the enzyme. So what the restriction endonuclease will do is that it is going to cut out the desired DNA piece, the desired DNA section, the section of the donor DNA that has the desired character. So it is going to cut it out. So once it is out, we can now do some things into it that will get it into the final cell. So Indamola, from what this particular section that I've explained, is it clear to you what we are doing with the donor DNA? Do you understand it now?
Yes, sir.
Now, so we have the desired DNA piece. So that one, let's hold it. But we cannot just throw it in like that into the cell of the organism that we want that character to be manifested. So we need the means of transportation because the DNA segment on its own will not get into the cell on its own. It needs something to take it inside it. And the way the means of transportation in this case, we call them vectors. Not vector the wrapper, so vector. Vector is the name that is used to describe it. And a good example, and it's called a plasmid DNA. A plasmid DNA can be used to deliver this package. It's just like you've ordered something on Jumia, and you are in Ibadan. If I order something on Jumia, and that thing will be I'm in Ibadan, but they are bringing it from Lagos. It will not just disappear from Lagos and land in Ibadan. It has to be transported. So that means of transportation is provided by the plasmid DNA. But as you can see on the other side, the plasmid DNA itself is also intact. It is circled. So we need to cut it open, so that the desired DNA, the desired package that we want to send to Ibadan can be properly integrated, incorporated, and can be delivered to its destination. Oindamola, is it clear so far?
Yes, sir. Yes, sir.
Nathaniel, are you following me?
Yes, sir, I am, sir.
So now, see the plasma DNA as the transport service that is going to bring my that is going to bring my junior package to me in Ibadan. But you know, if my order, which is a box, is put on the top of the box of the car of the lorry that they will use to deliver it, it can get lost on the highway. So it has to be packed properly well into the car that is going to bring it to Ibadan to me. So that car, that transport system is the plasmid DNA. So for that, my package to be packed into the car, they have to open the door of the car so that they can put my package inside the car and it will now bring it to me in Ibadan. So the way they do that is by using they are still going to use the restriction endonuclease. Don't forget, we said the restriction endonuclease acts as scissors, so I can cut off the desired region. In this case, the restriction endonuclease in my transportation, in my e-commerce shopping analogy, is opening the door of the plasmid DNA so that my package can be properly placed in the car and it will now bring it to me in Ibadan. So when we have it, when the restriction and the nucleus has acted on the plasmid DNA, it becomes a cut plasmid DNA. As you can see, the region that the restriction and the nucleus has cut open is now open. So which means the door has been opened for us to now place our package inside. The package that we want to put in this cut plasmid DNA is the desired DNA piece. So that we've taken from the source, from the person that has this character. So we now have to package it into the plasmid DNA. Now look at the layer, the line of the desired DNA piece and the cut plasmid DNA. So they are now going to be joined together. As you can see, the cut plasmid DNA and the desired DNA piece, that is now going to give us the recombinant DNA. So the desired DNA piece is now going to occupy the space that has been created by the action of the restriction endonuclease on the plasmid DNA. So when we have that, you can now see that the resulting end product does not look like the original donor DNA. It does not look like the plasmid DNA. It is as if an entirely different DNA has been created. And that is the reason for the name recombinant DNA. Precious, are we together?
Yes, sir.
Are you following me?
Yes, sir. I don't really understand the last one.
Nathaniel, are you following me?
Yes, sir.
So can you explain to Precious again, please, so that I think I can get as many people on board following me as possible. Can you explain what you have explained to the area that I've explained to your colleagues so that they can also catch up?
Okay, you said the donor, the particular DNA you want to get from the donor, it is being carried by the restriction enzyme, which is the restriction endonuclease. So the desired DNA piece goes to the DNA which we are taking it to, and when it gets to the DNA which we are taking it to, where we are extracting it to, what now happens is it is being transported, and for it to be transported, we use the plasmid DNA. So using the plasmid DNA, the restriction and the nucleus cuts the particular DNA that we're extracting it to so that it can fit in. So after fitting in, it comes in a new form.
Yeah, so that was a good trial. So we have the donor DNA. Look at what you have on your screen. The red region is the region for the donor DNA. That is the region. The red region is where the gene that codes for the desired trait is located. But we need to cut it open. We need to bring it out. We are not putting the entire donor DNA into the recipient. No, it's just that area of interest, which is denoted in color pink. This area of interest that we want to take to the recipient. So what we are now doing is that we use the restriction endonuclease, the restriction enzyme, to cut open the donor DNA so that we can be able to remove that area of interest. So once we've removed it, we said we need a delivery mechanism, a mix of transporting the desired DNA into the recipient that we want to express, that we want it to start expressing that particular trait. So the means of delivery that we have is by using the plasmid DNA. Plasmid DNA is serving as transportation mechanism. So the plasmid DNA itself is circular, as you can see, it's circular, it's closed. But we need to open it up so that it can properly transport the region of interest the DNA, the DNA segment that we want, we can properly transfer it into the host cell. So what we do to also open up the plasmid DNA is the restriction endonuclease. So with the use of restriction endonuclease, we are able to cut open the plasmid DNA. So once we cut it open, then we can now insert the desired DNA piece into the cut plasmid DNA. And what allows us, once that happens, once the recombinant plasmid DNA, the regions, the area that has been cut off, as you can see, there is something that's like a flat line on both areas. So these areas are the ones that will attach themselves to the desired DNA piece, so that it is now, the recombinant DNA is now closed again. But the closing will not be done on its own. There is another enzyme that performs this function, and the name of that enzyme is called ligase. Ligase is the enzyme. While restriction endonuclease opens up the DNA, ligase closes up the DNA after recombination has happened. So once ligase has closed up the cut plasmid DNA that now contains the desired DNA fragment, that setup that you are looking at that now contains that pink section is called the recombinant DNA. So if we want to have the gene for height, if we want you to have height, so what we do is that the gene that codes for height, we cut it out from the cell that has that property. We introduce it into a cut plasmid, into a plasmid DNA. We ligase it, the ligase closes it up, and that becomes recombinant. So how do you now express that trait? How do you now express increase in height, the height trait? So that cut plasmid DNA that contains the desired DNA piece is now introduced into the cell of the host. Into the cell of the host that we want those manifestations, those characters to start manifesting. Once we've introduced it, then multiplication and replication will now start happening. And replication will now start happening. And that particular gene, because it is producing a product, genes encode specific products. So the product of that gene that was expressed in the donor DNA will now start expressing in the host DNA. As you can see, the recombinant DNA is introduced into host cells, and multiplication happens, and the selection, this particular region is selected, and it is now product of that DNA fragment is now coming out. We are seeing it manifesting. If the particular organism, for instance, in sickle cell disease, if we are trying to apply it in sickle cell disease, it simply means that the gene, we are going to trace the region of the abnormality where the mutation is located. We are going to take the normal person, the person that has the right genes. We are going to cut it open using the restriction endonuclease. We are going to do the same thing. We are going to replace it. So once we have the recombinant DNA, we can now introduce it into the host cell. And that particular recombinant DNA is going to be amplified, and it will start producing the product that we desire. If it's insulin, if the patient is not producing insulin, we take the gene that produces insulin from somebody that is normal, that is having the normal insulin production. We are going to introduce it into the cells of a person that does not have insulin, and the DNA that we've introduced will now start producing the protein, insulin protein, that is encoded by the gene. Nathaniel, do you understand? Is this chart okay to you?
Yes, sir.
Precious Eziri. You see that Precious is not on a duty post. Precious Eziri, this is the second time I've been sending you out. Precious Eziri. I just want to see how long it will take her for to join us. Precious Eziri, we are waiting for you. Precious Eziri,
Sir,
where were you?
sir,
Where did you go to?
no, sir, I'm here. No, sir, I'm
Why didn't you
hearing you.
answer the question that I said? Obviously, you guys
Sir,
are the
I'm
good.
here.
How many seconds did it
Sir,
take you
I'm
to
here.
answer?
I'm here. Hello. Sir?
You are here. Answer the question that I had.
Sir, it's
You
not
are
going
here.
to be. I'm here. I've been shouting, I'm here. I'm here. I'm here. Maybe it's the
Answer the
network,
question.
sir.
Answer the question.
Sir, I said I am here. I said you asked me that Precious is Ziri. And I said I am here. You were asking Nathaniel.
I answered the question now that I asked Nathaniel. So answer the question.
Sir?
Did you get it? Did you get my question? The question I asked Nathaniel. Did you get it?
I don't know, sir. Hello, sir.
I'm listening to you. I just want your colleague to see. She has disappeared. So you guys, please, these things are not hard if you are fully committed. If you are willing to do the hard work of reading, if you are calm, if you are following me, Ekele Obona, you keep going in and out. Are you with us?
So I'm here.
Did you understand in a nutshell how we carry out recombinant DNA technology?
Yes, sir.
So please and please, so that is what we do in recombinant DNA technology. So we take something from an organism that does not have that character, and we put it, and we take something from an organism that has a character, that particular trait that we desire, that particular feature, that particular character, and we put it, the gene that codes for it, we put it in an organism, in the genes, in the cells of an organism, that does not have that particular character. We deliver it via the use of plasmid DNA. Two enzymes are important: restriction endonuclease and ligase. Restriction endonuclease is used to cut open the DNA fragment. So after we introduce it into the cut plasmid, the transport mechanism, then it is filled up with the use of ligase. So, and the recombinant DNA is now introduced into the cell of the organism that we want that trait to be manifesting, and it will start expressing it. So what I just explained, I want Nathaniel to describe how we are going to use that, how we are going to theoretically, hypothetically explanation of how we are going to make a tomato that has soft skin to have hard skin. So give me a theoretical explanation of what we are going to do.
Sorry, the network was breaking. I didn't get the question very well.
So my question is this: using the explanation that I've given you guys on how recombinant DNA technology works, I want you to give me an hypothetical explanation of how we are going to use that measure for a tomato that has very soft skin now have hard skin that can stand processing and handling pressure. So give me a hypothetical theoretical explanation of what we are going to do. The rest of it should also be thinking of the explanation in the context of what I described, non-specific. I'm waiting for you.
So the tomato that has a hard skin. I don't really understand the question. I don't understand the question.
We want a
Is
tomato
it,
that has soft. We want a tomato that has soft skin to have hard skin.
okay,
What do we do?
okay, okay. So the tomato that has soft skin, if we want it to have a hard skin, what we do is we um we take that uh particular gene from the tomatoes that has the hard skin, and then we take it it is taken by the restriction enzyme, which is the um, restriction endonuclease. It is cut out with so when cut out the restriction enzyme, that particular
Oindamola, can you take it from there?
again with also the tomato with the soft skin, and then it attaches with it, and then the, the
We are struggling to hear you, Nathaniel. So let Oindamola continue. Oindamola, can you take it? Can you answer the question?
the mechanism. Now,
Sure.
So please answer the
Okay,
question.
sir. Sir. Okay. The when the desired DNA piece. Oh no, the desired DNA piece from the hard tomato will be joined into the um soft tomato by enzyme called ligase. So that's where it stopped.
So if you are the one, if you give yourself that explanation, will you give yourself A or B or
No,
C or
I said,
or
so I said I should continue from where it stopped.
say your own? That's what I mean. Let me
Oh,
take it from
okay.
the top.
Um, the hard tomato we look the gene we look for the gene we're looking for. We use restriction enzyme to cut the particular gene we are in need of. Then the same restriction enzyme will be used to cut open um a part in the soft tomato. Then the the DNA that we've cut out from the hard tomato will be transported into the soft tomato through plasmid and joined to the soft tomato with through enzyme called ligase. That that's that's what I know.
So I will still have marks. You've only prepared the recombinant DNA. You've not introduced it into the host cell. After those manipulations that you've done, we are now going to introduce the recombinant DNA
Into
into the
the
host so that those proteins that the recombinant DNA encodes can now be produced. Is that clear, Yedamala?
yes, sir.
So now, so we've prepared the recombinant DNA, we've introduced it. So another thing that we need to mention today, because I'm really, really careful about overloading you guys here, but it's important that you get these things right, is, uh, is the relationship, is a concept that is called gene cloning. So when we've made our recombinant DNA, for us to be sure that this character is not present, we've only made one. We need it to produce in more copies. So that it won't be just one cell that contains this thing that we have. So that is where gene cloning comes in. So the goal of gene cloning is for us to get enough copies of the gene to manipulate. We are not trying to say. Let me use the example of what they are currently doing with COVID-19 testing using PCR. The PCR machine, the PCR technology is, I see it, I describe it as the simplest way for you to understand what gene cloning is. For instance, if something, so what they do is that the goal of cloning is that if we can get just something that is small, for us to be able to know whether that thing is present or not, is that if it contains even the minutest amount of that particular substance, we are now going to amplify that particular product so that we have it in large quantity and we will now have enough quantity of it for us to manipulate and work on. So gene cloning is a process through which we produce more copies of a particular gene so that we can be able to manipulate it. So that recombinant, that particular DNA of interest, we manipulate it, we mass produce it so that we can now have enough copies of it to manipulate. Precious, do you understand that definition?
Yes, sir.
Precious, okay, great. So the goal of gene cloning is to get enough copies of the gene so
Yes,
that we can
sir.
manipulate. If it's just one
Sir,
copy,
can you?
I've heard
Sir,
you.
I'm saying
So
yes.
thank okay. Thank you. So the goal of gene cloning is that if it's just one copy of the DNA of the particular gene that we could get, once we work on that particular copy, that is it. But if we want that particular copy, we want to maximize, we want to mass produce it so that we, even if we've exhausted this one, we can always have additional copies of that specific gene to work with. So that is the reason why we need that we have gene cloning. And everything that we're mentioning is all of them, they are interlinked. For instance, we have the gene of interest. So we have the cloning vector, which is where it will be for us to mass produce it before we have it. Then we have the recombinant DNA. So there is a vector that is going to that will be the place that the mass production will be. So the recombinant DNA is coming in. We're introducing it into the cloning vector, and so after that it goes into the host in which it multiplies. And so the what we're essentially doing is that in recombinant DNA technology, we are starting with just very few copies of a particular gene. We have just few copies of it, but in the end, we are having many copies of it. So the key element, the goal is like the other the now one section of the presentation is to let you know what we are trying to achieve when we do gene cloning. There is this, we've had the recombinant DNA. Don't forget that recombinant DNA that if you remember how we did it, the recombinant DNA that has the plasmid DNA and the DNA of interest. So we now want to have more copies of that. For instance, if you want to introduce a prepared multiple organism that will have it, so you can now, so that there is mass production of it. So this is where the gene cloning comes in. So we have the recombinant DNA that is introduced into the cloning vector. The gene is also introduced there. So when you have it together, we are now packaging it into the host cell. So once this has been delivered into the host cell, it is multiplied and we now have abundant copies of it. So in the essence, what we start with a few copies, we are now ending with many copies of that particular gene. So in a nutshell, that is what I want you guys to understand.
Excuse me, sir, I can't hear you. Hello, sir, I can't hear you, sir.
Nathaniel, can you hear me? Are you hearing me loud and clear?
So at the point I was hearing you, but it became not audible. I was struggling to hear.
So did you hear how I described gene cloning?
Yes, sir. You said the gene cloning it starts with it starts with a single gene. Then at the end it multiplies, it becomes many.
So what I said was that yes, you are quite right. Um, See gene cloning as the amplification of the recombinant DNA. What we are trying to do is that we are trying to make more copies of it. When we have more copies of it, of the recombinant DNA, then we can deliver it into additional cells. We don't have to go through the process all over again per organism. So we've been able to achieve the recombinant DNA. What we're now doing is that we are now mass producing that recombinant DNA so that we can have multiple copies of it. So the goal of gene cloning is that we want to have enough copies of the gene to manipulate so that we can work on the gene as much as possible. So what do? We have a gene, we have a recombinant DNA, we package both of them into the cloning vector, and this is delivered into the host. And when it is in the host, the cell multiplication system of the host will be producing more copies of that gene, and it is producing more copies of the DNA. Precious, why are you struggling to hear me? Nathaniel, did you hear me?
Yes, sir, I heard you.
Precious, can you hear me now? Sister Precious. Precious has left us. So, Oyedamola, can you describe in a nutshell what we are trying to do with gene cloning? Oyedamola, I'm listening to you.
Okay. So he said the goal of gene cloning is to multiply copy of DNA that starts with a single copy of DNA, then ends with many copies.
Yeah, in a nutshell,
It's
that's
also
the. Okay, continue, please.
it also helps to get enough copy of gene to manipulate.
So
And
why do you think we need enough copies? Why do we need more copies of a particular gene?
in in case so in case we are used we've used the I don't in case we use the first one and we need another one so we can go back to it. So I don't
So
you
yeah,
get
you are
it.
very correct. The goal of the process of gene cloning is that we want to get enough copies of a particular gene for manipulation. So it's just mass production. They look exactly alike. They are the same. There is no difference in them. And so we started with just few copies, very few copies. That's the starting point. By the end of the process, we are having many copies of the gene, and all of them are similar. They are identical to the starting gene. So all of them are identical to the starting gene. So they look the same. In all things because it's like we are making photocopies of what we are looking for. So they look the same, and there is no difference, and they're exact copies. So because we have exact copies, we can use them for different or multitude of functions. So the last thing that I don't forget, the goal of this class is to introduce you guys to some concepts. The next one I'm asked to introduce you to is called genetic marker. So genetic markers, by definition, is a gene or a DNA sequence with a known location on a chromosome and can be used to identify individuals or species. Let's say that definition again. A genetic marker is a gene or DNA sequence with a known location on a chromosome that can be used to identify individuals or species. So when if we want to differentiate between a man and a woman, as a human being, there are stuff that you look at uh, from many things. Sometimes some people will say that, okay, no, that is not the voice of a lady. That is how a man talks. In many cases, you can be right. Even when a man is standing in front of you and a woman is standing in front of you, you can easily know which one is a man or which one is a woman based on a combination of features and characters. But if you are presented with samples, if you are presented with DNA samples, and they are saying that, okay, samples, which of these DNA samples is gotten from human beings, and which of them is from a monkey, this is where markers come in place. This is where we now deploy the use of genetic markers. So that because we know genetic markers work on the principle that this particular gene, this particular DNA sequence is peculiar to this organism, or is peculiar to this species, or is peculiar to this organism. Nathaniel, speak. Can you hear me?
Sir, I couldn't hear you before, but I just hear you now.
Am I audible now?
You are.
Oyidamola, can you hear me properly?
Yes, sir.
So what I'm describing is a genetic marker. So I said a genetic marker is a gene or DNA sequence with a known location on a chromosome that can be used to identify individuals or species. So I now use the example of you guys now. Even though I'm not even looking at your names, I don't have all the features of you guys or details of you guys in my interface there. But I can say that from the voices of Oindamola and Nathaniel, I can say that Oindamola is a lady, while Nathaniel is a guy. So I can say that because we are used to hearing guys speak and ladies speak. And I can also say that if the two of you are standing in front of me, without asking for your cytogenetic gender, I can say that this is the man and this is the woman. But when I'm presented with your DNA samples and I'm not given any information about which is which, what I can use as a tool to identify who is who is to use genetic markers, because genetic markers will, specific depending on what you are trying to do. If you are trying to identify gender, you will use a genetic marker that is only found. You will look for a DNA sequence that is only found in that type of gender. If you are trying to distinguish between gender, a male or female, although the easiest way to do it in terms of gender is to look for the Y chromosome, because women don't have the Y chromosome. It's only the men that has Y chromosome. So you can conclude whoever has the Y chromosome is a male, whoever has two X chromosomes, whoever lacks Y chromosome but has two X chromosomes is a female. In species too, I said if you are given the sample of a monkey and a sample of a male of a human being, DNA sample, they will look alike in the container that they are going to come in. But the way you can identify them is for you to look for a particular DNA sequence that is only found in that species. If a particular DNA sequence is only found in human beings, that is the DNA sequence that you are going to look for to identify. So that is how we can differentiate between individuals and other species. And what we use is that it can be a short sequence. It's usually a very, very short sequence, not an entire chromosome, because for us to be able to amplify, it's easier to amplify a short sequence because we can quickly design a short primer that would only bind to that particular short sequence and can tell us whether what we are looking for is present or not. They use this for gene mapping. For instance, if you've seen the genetic map, the complete details and location of individual genes, because we have a DNA marker, we have the genetic marker that are peculiar to the various regions of the gene, of the chromosomes, of the DNA. That is how we are able to localize the various DNAs, and we've now been able to build a map around it. But because of what I'm getting, I'm not sure you guys can quickly grab in this introductory class what gene mapping is. But I think in an application that I can use for you guys is we can use it to describe relationships. We can use it to identify the genetic causes of diseases. For instance, if a patient has breast cancer, there is this BRCA1 gene and BRCA2 genes. They are the ones that are found to have mutation. So there is a specific mutation in the BRCA1 gene or BRCA2 gene that is strongly associated with breast cancer. So the mutation, the regions that we are expected to see that mutation is going to serve as our genetic marker. So what we are now going to do is that we've known what the mutation that is strongly associated with breast cancer looks like. So that particular mutation, we are now going to look for it. We are now going to screen for it. We are now going to attempt to amplify it, to clone it, so that if it is now present, we can say that this patient has this disease or not. So that is how we use markers. That is how we use genetic markers in disease diagnosis. So in disease prediction too, that even if a patient does not have breast cancer now, but she already has that mutant, that mutation, so that particular marker is going to tell us that this patient has a higher chance of suffering from this disease or not. Then another application that we use it for is in a major popular application is in resolving paternity disputes. So when you screen for when you identify, you look for peculiar DNA sequences that are found in the individual, in the prospective fathers, then you are now going to look for that particular peculiar gene in the genome of the baby, so that for you to be able to say that this is the father of the baby or not. So a short assignment that I'm going to give you guys is for you to describe how we use genetic markers in resolving paternity disputes. So let it be handwritten, let it be short and direct, and I want to have the results, the assignment today. So this is going to serve as your test, as your assignment. Please turn it in today. I want it to be handwritten, please. You write it out and you scan it and upload it in the Google Classroom. So the time is now 12 noon. So please and please, I will give you guys to 3 p.m. to turn in the assignment. How we use genetic markers to resolve paternity disputes. Handwritten, not typed, and I want it to be submitted in the Google Classroom page. So you upload it on the page, and I'm going to score it. So in a nutshell, today we've talked about recombinant DNA technology, gene cloning, and genetic markers. So we said when there is a desired character that you want in a person, in an organism, you introduce that particular character, you first excite it by using, you remove it, you take it out by the use of enzymes that are called restriction endonuclease. Then you attach it, you incorporate it into a cut plasmid DNA, and you seal everything up with the use of the ligase. Then you introduce, that forms of a recombinant DNA that is inserted into the host genome, that is inserted into the cells of the host cell. And from there it amplifies and it starts producing the products that we desire, and the trait starts manifesting. And we now said, even though we can do this process for few genes, we can actually have additional and more copies of it through the use of the procedure that is called gene cloning. In gene cloning, we are amplifying this particular gene so that we can have more copies of that gene to work with, and subsequently. And the products of gene cloning, they are exactly the type, they are exact, they are the exact type of the ones that we started with. But the end goal is for us to amplify it. And we have described genetic markers as short DNA sequences that are peculiar to individuals or species. And we said with these particular conditions, with the use of genetic markers, we can identify who is who, we can identify individuals, differentiate between gender, differentiate between species, and we also say that it is also used in cancer diagnosis, even if a patient does not have symptoms, because we have some mutations that are strongly associated with specific cancers. And these particular mutations have their whole sequences. So once we are able to do that, it enables us to identify a patient that has a stronger chance of developing a particular cancer. And I also told you guys to read up and submit an assignment by 3 p.m. today on how we use genetic markers to resolve paternity disputes. Do you have any question for me today, please? So, Ekele Ogbonna,
Yes,
When
sir.
So
you were saying about assignment,
so Nathaniel, what did you say?
no, I was to talk about the attendance. I have not taken attendance. I heard the link, something
I
about it.
sent a link. You guys should have been writing your names on the link. I sent a link. Only Fatbola Oyedamola has written her name. Why did the rest of you refuse to say it?
I've not seen the link, sir.
Because you came in late. Did you start with us?
Yes, sir.
How did Fatbola write it? Okay, so this is I'm putting the link in the group now. So write your name and matric number under a pad on that particular Google document. So I'm going to mark them and score them appropriately. Please make sure you can access it right now. Go and do it right away. Damola, did you see the link that I just dropped?
Yes, I've seen it, sir.
Ekele Ogbonna, did you see it?
Yes,
Ekele
sir.
Ogbonna. So Nathaniel, have you seen it too? Everybody, you should have seen it.
Yes, sir. I've seen it, sir.
So please write your name and matric number properly. Well, if you don't do it in the next 3 minutes, I'm not going to give you access to it.
So I saw it.
So please, and please do it right now. And yeah, so I'm going to. The time is 12:05. Immediately it is 12:10. You will not be able to access that link again. So do it right away. Enter your name, matric number, and don't be deleting each other's names. One of you should do the other will be for you so that you are not writing on the same line.
Sir,